Today, we are opening up the cell's microscopic toolbox. We're going to see one of its most critical jobs in action, controlling its own machinery. Yep, we're diving into the amazing world of enzyme regulation, the rules that bring perfect order to the beautiful chaos inside every single living thing. I mean, just think about it for a second. Inside every single one of your cells right now, thousands of chemical reactions are firing off. It's like a city that never sleeps, just on a molecular scale. So how does it all stay organized? How does the cell avoid total gridlock, wasted energy, and a complete system meltdown? Well, the answer is an incredibly sophisticated molecular toolkit. See, the cell doesn't just let its chemical reactions run wild. Oh no, it manages them with unbelievable precision. And today, we're going to pop the hood on that toolkit and see exactly how these little machines are kept perfectly in line. Okay, first things first. Before we can figure out how to control a machine, we gotta read the operating manual, right? So let's quickly cover the basic performance specs for our key players, the enzymes. The performance of most enzymes really boils down to two key numbers. First, you've got Vmax. Think of this as the enzyme's red line. It's absolute top speed, the fastest it can possibly work when it's completely flooded with fuel, or what we call substrate. Then there's Km. This spec tells you how responsive the enzyme is. It's the amount of fuel you need to get the engine running at half speed. So a really low Km means that enzyme is super efficient. It gets right to work even when fuel is scarce. All right, now that we know the specs, let's see how the cell slows things down. To do this, it uses a set of molecular brakes called inhibitors. We're going to look at three main types, and trust me, each one works in a slightly different and really clever way. First up, we have competitive inhibition, and you can think of this one like a game of musical chairs. The substrate, that's the molecule the enzyme is supposed to be working on, is trying to grab a seat at a special spot called the active site. But the competitive inhibitor molecule looks just similar enough that it can sneak in and steal that seat, blocking the real substrate from binding. It's a head-to-head race for the exact same spot. So what happens because of this competition? Well, interestingly, the enzyme's top speed, its Vmax, doesn't actually change. Now, why is that? It's because if you just flood the system with enough of the real substrate, you can basically overwhelm the inhibitor. By sheer numbers, the substrate will eventually win the race, and the enzyme can reach its top speed. But its responsiveness? That gets worse. The Km goes up, meaning you need way more substrate just to get the enzyme to half speed. Now, our second type of brake is called noncompetitive inhibition. This one's a little sneakier. The inhibitor doesn't even try to compete for the active site. Instead, it binds to a completely different location called an allosteric site. And when it binds there, it acts like a clamp, causing the whole enzyme to change its shape just enough to make the active site less effective, even if the substrate isn't there. And this totally different mechanism has a completely different result. Because some of the enzyme molecules are essentially taken out of commission, the overall top speed, Vmax, has to go down. You just can't work as fast with fewer functional enzymes. But, and this is key, the responsiveness, the Km of the enzymes that are still working, is totally unchanged. Their ability to grab the substrate is just as good as it was before. And now for our third, and maybe the weirdest break of all, uncompetitive inhibition. This is a true molecular trap. You see, this inhibitor can't bind to the enzyme by itself. Nope, it has to wait until the substrate is already bound and locked in. Only then can the inhibitor swoop in, grab onto that complex, and lock the substrate in place, preventing the enzyme from ever finishing its job. The effect here is really unique. Because the inhibitor is literally pulling active enzyme substrate complexes out of the game, the top speed, Vmax, definitely goes down. But here's the twist. By locking the substrate onto the enzyme, it makes it look like the enzyme has a much higher affinity for it. So its apparent responsiveness, the Km, actually decreases. Both the top speed and that responsiveness metric get lower. Okay, so we have three different molecular brakes, and each one messes with the enzyme's performance in a very specific way. But how on earth can a scientist tell them apart in a real experiment? Well, for that, they use a kind of special diagnostic screen for enzymes. It's called a Lineweaver-Burk plot, and it's just a really clever way of graphing all that performance data that turns the effect of each type of inhibitor into a crystal clear visual signature. It's a graph that tells you instantly which kind of brake is being applied to the system. So for competitive inhibition, the signature is a set of lines that all pivot around the same exact point on the vertical y-axis. That shared intercept is the giveaway. It shows us that Vmax, the top speed, hasn't changed. The lines just get steeper and steeper as you add more inhibitor, which shows that you need more substrate to get the job done.
Now, check out the signature for non-competitive inhibition. It's a completely different picture. Here, all the lines intersect on the horizontal x-axis instead. This tells us the Km, that responsiveness, is the thing that's unchanged. And as the inhibitor lowers the top speed, the lines just pivot upwards, raising their y-intercepts.
And finally, the signature for uncompetitive inhibition. This one is the most striking of them all. You get a series of perfectly parallel lines. You can't miss it. This clean, parallel pattern is the visual confirmation that both Vmax and Km are decreasing in lockstep, which is the unique calling card of that molecular trap. So far, we've been talking about hitting the brakes, slowing things down or stopping them. But the cell's toolkit is way more sophisticated than just a simple on-off switch. It also has dimmer switches for really precise, fine-tuned control. This fine-tuning is called allosteric regulation. And just like we saw with non-competitive inhibition, it involves molecules binding to that secondary, allosteric site. But here's the big difference. These molecules, which we call effectors, don't just shut things down. They can either turn the enzyme's activity up or dial it down. They act just like a sensitive volume knob. You can see the difference clear as day when you look at the response curves. A standard enzyme just turns on and ramps up to its max speed. But an allosteric enzyme, it often has this characteristic S-shaped curve. And that S is so important because it means the enzyme is incredibly sensitive to tiny changes right in the middle of its operating range. It's a true dimmer switch, allowing for incredibly precise adjustments. So let's put this all together now. The cell has brakes and it has dimmer switches. So how does it use them to create a fully automated, self-regulating system? For that, we turn to one of the most elegant designs in all of biology. It's called feedback inhibition. And the idea is as simple as it is brilliant. In a long biochemical assembly line, the final product acts as the signal to shut down the very first step of that line. It's kind of like a factory that automatically pauses production when the warehouse gets full of finished goods. It prevents waste, it prevents overproduction, it's just smart. Here's how the loop works. Enzyme 1 kicks off the assembly line. The pathway chugs along, and the final product gets made. But when enough of that product starts to build up, it actually travels all the way back to the beginning and binds to an allosteric site on that very first enzyme, acting as an inhibitor. This automatically pauses the whole pathway. And then, once the cell uses up the product, the inhibitor just floats away, and the assembly line starts right back up. It's a beautiful, self-correcting system. And this brings us to one last really critical question. We've just explored the cell's own toolkit for controlling itself. These binding sites, these allosteric sites, they're the natural switches and dials that life itself uses to maintain balance. So, can we learn to flip these same switches? How can we design drugs that specifically target these mechanisms to fix pathways that have gone wrong in diseases? Answering that question, well, that's the foundation of so much of modern medicine.